Good afternoon to all. In previous lecture, we discussed about the reproduction and their reproduction type, that is a vegetative, asexual, and sexual reproductions. Now, in this lecture, we discuss about the structure of the spores. We know that there are two types of the spores, that is microspores and megaspores, and how will they forms. Okay. नाव आज जर आपण दोन लेक्चर घेणार आहोत. एक लेक्चर असेल ते 12:58 ते 1:46 आणि दुसरं असेल ते सायंकाळी 6 ते 7च्या दरम्यानमध्ये. ओके, आज दोन लेक्चर असतील ते सर्वांना लक्षात ठेवा. पाठीमागच्या लेक्चरमध्ये आपण स्पोर बघितले, त्याचे टाईप बघितले. ओके, त्याची पोझिशन बघितली, हाऊ कोन विल द फॉर्म, हाऊ स्पोरोफिल विल द फॉर्म, what is the meaning of the vegetative reproductions. Okay, there are various types of their fragmentation by the tuber formation, that is adventitious birds. Then we see the asexual reproduction. That asexual reproduction is take place by the formation of the spores. The spores are the haploid. Okay, there are two type of the spores. When plant is heterospores, they form two type of spores, that is the microspores, and second is the megaspores. Microspores are form in the large number, as well as the megaspores form in the 1 to 4 numbers. Okay, and they having the larger size than the microspores. Now we can actually see how these microspores forms. Okay, their structure, how they look like. Okay, microspore and megaspores. Then first is the mature microsporangia. Now in this lecture we see the actual structure of the microsporangium as well as the microspores and their process formations. The first points regarding to these mature microsporangiums. Okay, the microsporangium is a short stalked structure. Okay, you know that the microspores are formed inside the microsporangiums, and these microsporangiums are present on the microsporophyll. This is the basic, and you know that. Now first point is the structure of the microsporangium. The microsporangium is short stalk structure. This is a short stalk structure. This is having a very short stalk. Okay, the length having very short, therefore we call as the short stalk structure with two layered sporangial jacket. These having two layer of the sporangia. This is the two layered jacket with two layered sporangial jacket. Now here we can see this is the structure of the microsporangiums. Okay, we can see that here the very short stalks are present. This is the stalk, this is are very short. Then this microsporangium having two layered structure. This is the first layer, this is the second layer. There are two. Okay, the two layer, first is the stalk, then first layer and second layer. Then inside there microspores are present. You can see that here the microspores are present. This is the microsporangium having two layered. Then ligules are present. This is the ligules, and whole structure is this whole structure is known as the from the ligules and these cells, and this is called as the microsporophyll. This is the structure of the microspores. Okay, this is structure of the microspores. This having two wall. Outer wall is known as the exines, inner wall is known as the entines. Exine wall is a thick and entine wall is a thin. Exhale wall having the ornaments. Okay, they are having the ornamentation on their outer wall, where thin wall is not having ornamented. This having outer wall consists of the triradiate region. This is the triradiate region. This having the triradiate regions. Likewise, this is the important the structure. This having the stalk, this having the ligules, this having the microsporangium. This is the microsporangium. This microsporangium are two layered structures. Then inside that the microspore cells are present. As you see there inside this microspore, this having the nucleus. So this having the nucleus, and this nucleus is surrounded by the cytoplasms, and this cytoplasm also having the reserve food material in the forms of oil globules. This is the about the structure of the microsporangium and spores. We can see here figure 4.1 mature structure of the microsporangium and microspore structure. Now this diagram is clear. Then we move on theory. This cell of the outer jacket, the cell of this outer jacket contain chloroplast. These cells, this cell is the outer chloroplast. This contain chloroplast. It consists of short stalks. This microsporangium having short stalks, we already know that, and oval. This shape is the oval shape. Reniforms or spherical capsule. This is looking like the spherical carpule with the short stalks, and two layered structure. Okay, then next point, that is a microsporangia. Microsporangia are usually slightly elongated. As you see here, these microsporangia, these are the slightly elongated structure. The microsporangia are usually slightly elongated, yellow to red or yellow to red orange in color. They also having microsporangias possesses their own color, that is the having yellow to red orange in color. Subtended by a small, these are subtended by a small flap-like ligule. We can see that here the ligules, a flap-like structure. Okay, this is a tongue-like or flap-like structure. It is known as the ligules. Okay, subtended by a small flap-like ligule at the adaxial surface. Microsporangium. This microsporangium contain microspores with germinate inside the microsporangium forming male gametes. Now here is important concept. We remember that for your lifetime. That is a microsporangium. This microsporangium contain microspores. Okay. Now this terms clear. The microsporangium contain microspores. These microspores, when get matured, they start germination inside the microsporangium. These microspores, this is a very important concept, that is the microspores start get germinated inside the microsporangium when they get the accurate maturity. When they get the maturity, they start germinate inside the microsporangium. That is the microspores contain microspores which germinate inside the microsporangium, and then they form the male gametes. When they release from this the microsporangium, they form the male gamete. We know that the microsporangium from the male gamete, megasporangium from the female gamete. Okay, this point is clear. At the distal end, here we can see that the two end, distal end and this is the distal end and this is the proximal end. At the distal end, a line of dehiscence. We can see that here this is a line of dehiscence. At the distal end, a line of dehiscence is present. Here the line of dehiscence is the present, through which it dehisces transversely, and these lines of the dehiscence dehisce transversely and to the axis of the sporophyll. This is the axis of the sporophyll, and at the axis of the sporophyll, the line of dehiscence is present, and this line is the dehiscence. Okay, this is some hygroscopic change take place in the outer layers of the sporangiums. This hygrospore changes inside the layer causes the dehiscence of this microsporangium. Then the spores will be the get released, and when they fall down on the soils or substrate or surface, when the condition is the favorable, that is plenty amount of water moisture is available, they get germinate. Okay, and from the male gametophyte. Then next, okay, mature. Now this structure of the microsporangium is clear. Then now we can see the structure of the microspores. Here we can see this is the microspores. This is the microspores. The microspores are very small in size. As you see that the size of the microspores is very small. This is a very few micron. Okay, we cannot see by your naked eye. We require the microspore for see the structure of the microspores. Okay, these microspores are very small in size. Next point, outer wall is thick ornamented. Then outer wall, this having the two layer, that is outer wall and inner wall. The outer wall is thick. The outer wall, this is the outer wall of the microspores. This outer wall is a thick ornamented. This is ornamented. You can see that the specially design present on the outer wall. This is called as the ornamentations. Okay, outer wall is a thick ornamented, known as the exhine. Outer wall layer is known as the exhine. Inner layer is known as the entines. Okay, outer wall, this is exhines which are the ornamented and having a thick, and inner wall is also known as the entine. This is the outer layer, it is known as exhines. Inside there the inner wall, is known as the entines. The next point, each spore has a single nucleus. As you know that I show that there is each this is the microspores. Inside that the nucleus is present. Inside the nucleus DNA is present. Okay, this is the controller head of the cell nucleus is known as Each spore has a single nucleus surrounded by a cytoplasm. This is surrounded by the cytoplasm. It contains a reserve food material in the form of oil globules. These microspores contains reserve food material in the form of oil globules and nitrogenous material. These microspores consist of the cytoplasm. Inside the cytoplasm, reserve food material is present in the form of oil globule as well as they having the nitrogenous materials. Okay, these are the required for their germination during the germinations. Then exine has a distinct triradiate mark. As you see, you have the exines having these triradiate mark. This is the exines and they are having the trimadiate tri. From this triradiate mark, the germ tube comes. Okay, and then the male gametophyte will be the form. This is about the microsporangium. This is a structure and microsporangium. Okay, we can revise very fastly. These microsporangiums are formed on the microsporophylls. Okay, this microsporangium having a very short stalk, and then this having a double wall layered jacket. Okay, then inside this microsporangium, microspores are present. Okay, at the axil of the each microsporophyll, there is a dehiscence. Dehiscence take place and microspores get released. Structure of the microspores, as you see, the microspores having two wall. Outer wall it is known as exine, inner wall is known as the entine. Exine is the ornamental and thick, while the entine is the thin walled structures. Okay, this is about the structure of the microsporangium and structure of the microspores. Now in next lecture we see the structure of the megasporangium and structure of the megaspores. We can stop here.